Good morning, everyone. Thank you for joining the session at ACDLabs Virtual Symposium. My name is Yu Hui. I am a scientist in Discovery Chemistry Department at Genentech. I'm located currently in South San Francisco. It is a great pleasure to be here today to discuss a new tool we developed in collaboration with ACDLabs to streamline our quantitative NMR workflow. In this presentation, I will walk you through two key components of this project. The first one is we try to implement a robust system for external standards, and the second one is how we leverage that external standard system to build a digital workflow. So to set the stage why QNMR process is so important, let's first look at the typical workflow for new compound registration in discovery chemistry here at Genentech. It all begins on the left where our scientists and medicinal chemistry group and biology project teams design novel molecules for the targets we have in-house. The synthesis is then either performed in-house or by our partners at contract research organizations. All the synthetic compounds then move into the second step, purification. This is handled by a dedicated group of scientists in our purification group. Those purification experts typically run a MS-directed purification using SFC or HPLC as the picture shown in the middle here. This purified compounds then go through QC before they getting registered to the compound management. For small molecule QC, we focus on identification and purity. We typically run LC-MS and 1D and 2D NMR experiments on all of our QC compounds. You can see in the picture on the left here, it's a LC-MS instrument we typically use. It's a thermal QE. We use it to run the regular UV, CAD, and SPEC. And on the right side is what we use for our NMR analysis. We have 3 magnets in total, which we can use either with walk-up or research. Some of the key aspects we look for during QC is purity, either by UV or CAD, molecular formula match by high-resolution mass spec, and NMR correlation that helps us to connect the structure. To date, we maintain a turnaround time of 1 day for small molecule registration QC. A critical part of our analytical workflow isn't just generating high-quality data, but also managing it effectively so it can be easily assessed and leveraged for future projects. To solve this, we partnered with ACDLabs to build a comprehensive and fully searchable analytical database. This has been a significant undertaking, and the database now houses over 15,000 records that include both NMR and LC-MS records. Beyond simple storage, the database is an active tool. It allows us to generate purity report with different detector, and it features powerful search capacities. Our chemists can instantly find data not just by a compound ID, but also by searching for chemical substructure. It is very useful to exploring related analogs. Depending on where the compound is moving towards, we decide what experiment and assays we need to run. If the compound is moving toward a routine bioassay, like checking if it is more potent, if it has a better PK, or if the SAR meets our expectation, we typically run LC-MS experiment and 1D, 2D NMR to register the compound and get the compound into the assay. As shown in the form here, it takes about 1 milligram of material, and we get the results back to the chemist in 1 day. If the compound is moving into an animal study, the stakes are much higher, so we need much more analytical scrutiny. We generate a certificate of testing report as shown on the right here. In those cases, we'll need about 25 milligram of material. We run a list of experiments, including NMR for structure ID, HPLC for organic impurities, Carl-Fisher for water content, Chiro-QC for enantiomers or stereoisomers, and QNMR is super important in this case because it can detect any impurities or inorganic salts that may not show up in all of the other technique. This helps us ensure potency or purity before the compound goes into any animal study. So having established the need for high quality quantification NMR, I want to talk about the method itself. Since 2024, our lab has fully transitioned from using internal standard calibration to using external standard calibration for all of our QNMR analysis. And there are three key benefits we saw. First, with external calibration standard, we were able to simplify our sample preparations. Instead of adding internal calibrants to every single sample, we typically run external standard on a weekly basis. More importantly, when we observe that when we mix the samples with standard, it may cause some unexpected interactions or even sometimes reactions. Using external standard can completely avoid that. If necessary, we can even recover the material. Thus, using external calibration standard improves sample prep efficiency. In addition, by applying solvent correction with external calibration approach, we saw an improvement in our precision. Our sample-to-sample variability dropped from nearly 3% down to less than 1%. Besides that, when we perform the analysis using 4 different peaks from the same sample, the average relative standard deviation is just over 1%. This tells us the results are consistent and reliable. Now let's take a moment to simply look at this mathematics. This equation is a foundation for external standard. Typically, a reference material with known concentration is used to determine the CCF value, which stands for concentration conversion factor. The CCF value then acts as a calibration constant, which we can analyze our unknown sample against and use that same factor to accurately calculate the unknowns, either concentration or potency. However, for this method to be reliable, we need to account for some experimental variations. Some salty samples, pulse pulse corrections could be useful. Usually, we'll use the 90-degree pulse length to normalize for probe tuning differences. In other words, pulse correction compensates for ionic strength difference between samples. And also, not all NMR tubes are made the same. We use solvent correction for variations in different NMR tubes. By implementing these corrections, we can be confident that our results are both accurate and reproducible. One of the biggest challenge, as I just said, with external standard was always difference between the standard tube and the sample tube, which we call it tube-to-tube variability. In this case, we use the residual solvent signal, which is present in every single sample as a built-in reference. Some people call this ECIC, external standard and internal standard method. On the screen, you can see the spectrum for both our analyte and our standard. In this case, is caffeine. They both show the DMSO peak at 2.5 ppm. By using this common solvent peak to normalize the data, we can compensate for the tube variability. This solvent correction approach is a key upgrade that improves the accuracy of our measurement. It almost eliminates the need for traditional internal standard approach. But having a validated validated method is not enough. We want to roll this out to the entire chemistry team in a standardized and scalable way. Our first solution was the Excel-based tool. This slide shows the main summary page. All the orange part are our inputs. Here, either an NMR expert or the bench chemist will need to enter all the critical sample information in the yellow cells. Things like the compound ID, its molecular weight, the exact sample weight, and the solvent volume used for dissolution. Based on those inputs and other data pasted in from the data processing software, the spreadsheet would then calculate the outputs shown here in blue. It lists the key results, including the final average weight percent from three separate preparations, the standard deviation, and the maximum percent RSD, and which tells us about the consistency of the measurement. The next step in our previous workflow was the standard information entry. This task is usually handled by an NMR expert. The experts will need to manually input all the standard matter information. For example, the DMS we use to dissolve the standard, the lot number of the standard, the weight, and the concentration. For this process, we use the certified QMR standard for 4-difluorobenzophenone because it works for both proton and fluorine QMR. For proton QMR, we use CCF as our concentration conversion factor without solvent correction, and the DCCF is used as the concentration conversion factor with solvent correction. Most importantly, we usually use at least 3, sometimes 5 replicates of standard. We do this for critical reasons. First, it gives a much more accurate and robust calibration, and second, it serves as a valuable check on the stability of the instrument over time. The final step in the Excel workflow was sample data entry. The chemists need to switch back and forth between NMR data software and QMR analysis Excel. In the NMR's processing software, the chemists need to run baseline correction, phase correction, zero filling, probably multiplet analysis. Then they need to switch back to the Excel to manually copy the pick list from the NMR software and then paste it into this Excel sheet. They would have to manually select the signal for quantification and label the solvent peaks. While the Excel workflow have a lot of benefits, it's easy to develop, and it's a great workaround for external calibration with multiple replicates. It ultimately have presented some challenges that we cannot neglect. First, it requires extensive chemist training, and second, there's a lot of manual process in this Excel format that may induce some human errors. And third, when we try to upgrade or update our calibration file, it's a little bit hard to manage that process. Last but not the least, this process is not automation ready. That is why we decided to search for a new software solution, one that can combine data processing and data analysis in the same place, and that can minimize human error, streamline the process, and prepare us for automation. With the transition to the new QMR tool developed by ACDLabs, it has allowed us to streamline our workflow in 3 steps. First, we built a robust calibration file using multiple replicates of external standard to ensure accuracy. Second, we directly import all sample information and spectral data, which eliminates the risk of manual copy and paste error. And finally, the tool provides automatic report generation, saving significant time and standardizing our outputs. The new workflow begins as before by building a robust calibration file from our standard replicates. We usually use at least 3, sometimes 5 replicates. The key advantage of the ACDLab plugin is the ability to use a macro for data processing, which you can see here on the left. The single macro automates all the critical steps, including data processing, phasing, baseline correction, and data analysis integration, applying the exact parameter to every single spectrum we have. This is a great combined processing and analysis template because it ensures the consistency and eliminates the potential for human bias that can occur during manual processing. This is the concentration calculation interface, which we built the calibration file. The solvent automatically read parameters like the number of scans and gain values directly from the FID files. It also auto-populates the standard weight and volume of the solvent used to dissolve the standard if we enter it in our parameter set on the NMR instrument. This applies to both Bruker and gel system. To ensure every spectrum is processed and analyzed the same, the software lets us define the integral regions on one replicate and then transfer that analysis across all other replicates. If pulse length-based correction was run, we can also define this pulse angles. And after every replicate looks good and looks the same, we can save that calibration file with multiple replicates of standard. When it comes to sample data processing using this QMR tool, in addition to the nice feature we mentioned before, like transferable multiplet analysis, auto reads and populates experimental parameters, we are also able to click on and off the solvent correction. We can dynamically select or deselect which peaks to include in the calculation. If we want to exclude an exchangeable proton, we just uncheck it, and the final weight-to-weight percent on the right will automatically recalculate. In addition, when we can also select different calibration files. For instance, we can select different solvent like chloroform or DMSO, or other nuclei like fluorine 19 NMR. Besides that, we will be able to see details on each replicate. Once all the data is cleaned up, and the software will automatically generate this report that can be exported as an PDF. Finally, let's take a look at the output. At the very top, the report provides the critical information. For instance, it gives the final purity as a weight-to-weight percentage. It also states the precision of the measurement through percent RSD or and maximum peak-to-peak RSD. This summary section gives chemists and the project stakeholder what they need quickly and unambiguously. In addition to the summary information, the first reporting page also includes information like using CCF and the detailed on each replicate. Here you can see the specific sample weights, volumes, and calculated purity for each of the 3 runs. This allows anyone reviewing the data to verify the consistency across the measurements and understand exactly how that final weight-to-weight percent was derived. The previous page gives us the final answer. This page is for analytical scientists like me. This is where we get to understand the detail of each replicate. First, on the left, you get the processed spectrum. You can see integrate peaks and confirm that that potential interference like solvent peaks and water peaks were correctly identified and labeled. Below is a table of integrals. It shows which signals were included in the final weight-to-weight percent, and this helps us to improve transparency. And finally, on the right side, gives us experimental details. Every single experimental parameters, for instance, those pose sequence we used or acquisition time we used, it is important because it helps us to replicate ourselves in the future. So to bring it all together, we began by looking at our previous Excel-based workflow. While functional, it was a manual process that was time-consuming and highly prone to human error and required extensive training for our chemists. By implementing the ACDLab QMR tool, we fundamentally transformed this process. As we've seen, the new tool provides a highly interactive interface where results update in real time, giving our scientists immediate feedbacks and complete control. By automating this data transfer and calculation, we can minimize the human error that is involved in this process, and it was a major liability in our old copy and paste method. The new workflow also provides streamlined calibration file management and is fully automation ready. It helps us to prepare for our future high throughput experiments, and most importantly, the simplified the chemist training, allow ourselves to spend more time on portfolio support work. All this work cannot be done without the support from my colleagues in small molecule drug discovery and ACDLabs. I would like to give a shout out to Mary, Ryan, and Dimitris, who spent a lot of time on this QMR tool, make sure it's easy to use and it's accurate. I would like to also thank you all for being here today. And with that, I would like to welcome any questions you may have.